Hello, everybody. In this lecture, we are going to go to the inside of the eukaryotic cell and talk about our intracellular structures. And let's get right into it. So, kind of like with the extracellular structures, we do have a lot of objectives. And so, to kind of go through it, by the end of this lecture, you should be able to describe the eukaryotic ribosomes, where they're located, explain the structure and function of the cytoskeleton, and name the organelle that builds microtubules. Describe the general structure and function of the nucleus, structural and functional features of the endoplasmic reticulum, and describe the general structural and functional features of the Golgi apparatus. Um, Talk about the different types of vesicles and vacuoles that are found in eukaryotic cells, and review the structure and function of mitochondria, and state how they're similar to bacteria. All righty. And so in our prokaryotic cells, we do have ribosomes, just like we do in our prokaryotic cells. And just like in prokaryotes, the function of the ribosome is to make proteins. And they are made primarily of protein and ribosomal RNA or rRNA. And so those rRNAs are known as ribozymes, or they're catalytic RNAs or enzymatic RNAs. So basically, they're enzyme or they're nucleic acids, specifically RNAs, that have the ability to have enzymatic function. And so the major difference between the eukaryotic and prokaryotic ribosome is that the eukaryotes have an 80S ribosome, whereas in the prokaryotes, it's a 70S ribosome. And here they are subunits of 60S and 40S. So the large subunit is 60S, the small subunit is 40S. And so what's important to know here is that these numbers are different, and there's some differences in the rRNAs as well. One of the things that is kind of interesting is what that S stands for. So I don't think I've mentioned this yet, but the S stands for Svedberg unit. And so a Svedberg unit is a measure of sedimentation rate. So the way that you figure this out is that you take your ribosomes and you spin them in an ultracentrifuge, and where they settle out in that centrifuge tube is the sedimentation rate. And so something that settles at 80S is going to settle below or further down than the 60S and 40S. And so that's kind of an interesting little way of measuring this thing. And that's why it's not additive. So it's when you have a 60S and 40S subunit, they do not equal to 100S because it's not based on like a weight or something like that. It's sedimentation in a solution. All right. And so when we make proteins in our eukaryotic cells, in our ribosomes, there are two places those ribosomes can be found. And the two places are going to be either free in the cytoplasm, and in these free floating ribosomes, they're going to make proteins that are going to primarily be produced or primarily be used inside of the cell. However, there are some ribosomes that can be bound, right? And those bound ribosomes are going to be bound to your endoplasmic reticulum, or the ER. We call this the rough ER because it has it's this endoplasmic reticulum with these little ribosomes embedded. And these are going to make proteins that are primarily going to be shuttled outside of the cell. Now, when you have ribosomes that are in the endoplasmic reticulum, or if they're in the cytoplasm, that doesn't mean they always have to be there. The cell can actually, change where ribosomes are primarily located based on the needs of the cell. And so we have the eukaryotic ribosome and the prokaryotic ribosome, which I explained, and keeping in mind, which we have mentioned before, that the prokaryotic ribosome is actually very similar to the ribosome of the mitochondria and chloroplasts. Those two organelles actually have their own ribosomes, made up of a 70S ribosome with a 50S and 30S subunit, as well as you've got some similar RNAs as well, like that 16S RNA. Now, the cytoskeleton in the cell is going to coordinate cell movement or cell cargo movement. So things that need to be transported in or out of the cell are going to use this cellular highway made up of this network of protein fibers. Now, there are several functions of the cytoskeleton. It helps maintain shape. This is very similar to the functions of the cytoskeleton in prokaryotes. It's going to facilitate movement. It's going to protect against external forces. So this is kind of adding on the benefits of having a cell wall. This cytoskeleton can also help. It helps transport both organelles, so it helps move organelles around the cell, as well as vesicles, which we'll talk about momentarily. And it's important for cell division. So when we're talking about mitosis and meiosis, the cytoskeleton is going to be very important there. Now, there are three main types of cytoskeleton fibers. We have our microtubules, intermediate filaments, and our microfilaments. Now, microtubules are these hollow tubes. They're made of tubulin. And this is when I said the highways of the cell, this is really what we're talking about here. This is going to include proteins like kinesin and dynein that move cargo along these microtubules. And during cell division, they actually form a spindle that helps separate the chromosomes, right? So the goal of the microtubule in general is to move things around the cell. And so during cell division, this is going to help move the chromosomes across the cell. Now, the microtubules arise from a centrosome. This is located near the nucleus, and it's made of these two centrioles. And so keep in mind that these centrioles are also the basal body that make up that eukaryotic flagella. Now, our intermediate filaments are more rope-like. They're a little bit smaller, and they can be made up of a host of different proteins. And what these do is they help contribute to the cell's strength. So it's tensile strength, its ability to withstand pressure. Our microfilaments are smaller. They're going to be made up of actin. They associate with myosin. So if you think of like a muscle contraction, they're also used for pinching the cell apart. And they can be used for movement for organisms that have a pseudopod. And so the microfilaments can be involved with that as well. Then we jump into the nucleus. This is where the DNA is housed. It's going to be organized in chromatin, and it's going to just float around in the nucleoplasm. This is different than the cytoplasm, which is kind of like the inside of the cell. The nucleoplasm is the inside of that nucleus. Now, the nucleolus is this dense area inside the nucleus. We can see it in here. It's where we've got all this stuff densely packed. It's going to be enriched with RNA, and this is going to be where ribosomal subunits begin their development. Now, your nucleus is enclosed by a double membrane structure called the nuclear envelope. It has nuclear pores, which allow things to traffic in and out of the nucleus. Now, your endoplasmic reticulum, which we can see down here, is going to be in continuous connection with your nuclear envelope at certain parts of it. So here we have our nucleus, looking at this SEM, this scanning electron microscope image. And then down here with these little folds, we have our endoplasmic reticulum, or ER. And so the ER has some pretty important roles for protein and lipid production. Many of the things that are being made in here, both proteins and lipids, are going to be they're going to be moved to other organelles, or they're going to be excreted out of the cell. These membranes is just really just this series of folded over membranes that originate at the side of the nuclear envelope, and they can be rough, which means that they have these ribosomes, or smooth, where they have no ribosomes. So the rough ER is going to be peppered with these little ribosomes on the outside. This is where these ribosomes are like directly inserted into the membrane. So it's similar to like your fluid mosaic idea, in that between the membrane you can have proteins. And here you're going to have protein modifications occurring, such as clips and folds, and even additions of organic and inorganic factors that can be added to those proteins. Kind of like when you add iron into hemoglobin. Now, your smooth ER is not associated with ribosomes. It's interlaced. It has a more tubular appearance. This is primarily used for lipid production, and it helps in detoxifying cellular toxins. And so we have the Golgi apparatus. This is going to be is going to be formed by vesicles that bud off from the endoplasmic reticulum. And they get shuttled over to this Golgi apparatus, which is going to be this plasma membrane here. And so these vesicles that we see on the ER are going to transport stuff over to your Golgi apparatus. The Golgi apparatus is going to be made up of this like cisternae, these disc-like flattened sacs that you see here. And they're going to be directly coordinating with the ER. And a lot of protein modification occurs here, some building of lipids, and it also helps send out stuff to where it's needed in the cell. And the things that do that sending off oftentimes are these vesicles. They are made up of a lipid bilayer. So you see here, we've got our hydrophilic head and our hydrophobic tails, and they are going to carry things inside of them. Sometimes when you're transport vesicles, they're going to move stuff around the cell. You have secretory vesicles or secretory vesicles that send things out to the cell surface that are going to be discharged from the cell. You have lysosomes, which contain these hydrolytic enzymes that can actually eat up organic matter and break down organic bonds. And then you have peroxisomes that contain these enzymes and also the ability to produce oxygen radicals. And they help protect cells from these toxic oxygen intermediates that are produced when oxidizing things. Vacuoles are like a bunch of vesicles that kind of merge together. They oftentimes just contain water or other nutrients or toxins. They can be various sizes. They're very common in plants and in fungi. And sometimes you get some special types in protists, like contractile vacuoles, which help regulate osmotic pressure for protists that don't have a cell wall, and keep them from lysing when they're in a hypotonic environment. Then we have our mitochondria and chloroplasts, which we spent a good amount of time talking about in the endosymbiosis lecture. But keep in mind, they have a circular chromosome, they have a double membrane structure, and they have that 70S ribosome, just like your prokaryotes do. So your mitochondria is going to be making your ATP. It also makes amino acids and vitamins. It regulates cell division, and it can even carry out apoptosis, or programmed cell death. It can be involved in several disease states, and play a role in both cancer and aging. Now, here is a cross-section of a mitochondria. You've got your smooth outer membrane and an inner membrane consisting of these cristae, right? And cells can have a different number of mitochondria depending on how active the cells are. And then only your photosynthetic cells are going to have chloroplasts. But most eukaryotic cells, including plant cells, have mitochondria, although there are some exceptions. So your chloroplasts allow the cell to harvest energy using sunlight. So this is where photosynthesis takes place. They're going to have some sort of light collecting pigment like chlorophyll, and that's how they absorb the light to fix carbon from the atmosphere and turn it into sugar. They also have the inner and outer membranes. They have these stroma rather, and these thylakoid membranes. And when they get stacked together, these are called grana, connected by the lamella. All right, so that's a lot of information about intracellular structures. You should now be able to describe the structure and function of the ribosomes and the cytoskeleton, the nucleus, the ER, and the Golgi apparatus. Explain what vesicles and vacuoles do, and review the structure and function of your mitochondria and chloroplasts. With that, thank you very much, and I'll see you soon.